the MHC molecule. And today we will be starting with the antigen processing and presentation. So T cells, just like we said, the MHC molecule represents the adaptive immunity, adaptive immune response of the body. Okay, and to do that, antigens or viruses or bacterial antigens or mutated cells have to be presented to this molecule before they can handle them or eliminate them from our body system. So we're going to look at the process, the means by which this antigen processing and presentation occurs. We say that T cell need presentation, okay? For T cell to recognize antigens, okay, proteins must be digested into peptides. Protein must be digested into peptides by antigen presenting cells. By antigen presenting cells. These people that are joining late are disturbing us, so I have to lock this meeting. Yeah, they're disturbing. Okay, so for T cells need presentation. Before T cells can recognize any antigen or foreign organism or viral infection to the body, the protein must be digested into peptide that is processed by antigen presenting cells. Okay, then antigen processing pathways, we have the immune system uses different pathways to associate peptides with MHC molecules and to transport the MHC peptide complex to the surface of the cell. Okay, we have the cytosolic pathway, that is the endogenous pathway. These bacterial infections are already inside of the cells are already infected. Okay, then we have the endocytosis pathway or the exogenous pathway, where the cell have to engulf the invader that the body wants to eliminate. So the mechanism for the major histocompatibility complex 1, the mechanism whereby it processes pathogens, viral infection, it processes it for presentation, just like we said, is through the endogenous pathway. The MHC class 1 glycoprotein presents antigen of endogenous origin to T cell receptors of CD8 cells. Okay, so for the class 1 molecule, what they do specifically, majorly, is to present antigens that are of endogenous origins, that is, antigens that have invaded the cell. Okay, so these endogenous peptides can be derived from the degradation of intracellular proteins, okay, intracellular bacteria, and mutated cell proteins, okay, the viral proteins, okay, that have invaded the cells. Okay, a small amount of these degradation products or proteins is directed to the proteasomes, in which these proteins are degraded into short peptides to be transferred to the endoplasmic reticulum, where they are locked on MHC class 1 molecule via the peptide loading complex. This is a complex that is involved in the movements of the peptides. Okay, so the cellular components involved in the presentation of all these cellular components involved in the presentation of this endogenous antigen from proteasome subunits to the peptide loading complex are collectively referred to as antigen processing machineries. Okay, so we'll look at the diagram, the next page, where we itemize it one by one. So the MHC molecule presents pathogens, antigens to CD8 cells. Okay, so first of all, proteins are proteolytically processed in the cytosol by the proteasomes in the cell. Then secondly, this is just the itemization of the steps involved in the processing and presentation of these antigens. Secondly, the peptide generated by the proteasomes are translocated into the endothelium reticulum lumen by TAP. Third, the MHC class 1 molecule, okay, fold and assemble in the ER lumen with the aid of the ER chaperones, carnesine, okay, car reticulum and ERP53. Okay, these are the molecules involved in the movement and processing of the antigens. Okay, the MHC class 1 molecules in a complex with car reticulum and ERP53 associate with TAP, and TAPsin facilitate peptide bonding. Okay, peptide-loaded MHC class 1 molecule dissociate from TAP and are transported through the secretory pathway to the plasma membrane. Antigen processing. So when you look at this diagram, you can see the target cells that have been infected already. Okay, so the processing that is happening in the endothelial reticulum, after which the antigen is being presented on this antigen presenting domain, antigen presenting groove of the MHC class 1 molecules. Okay, after the antigen had been processed, is being transported on this antigen presenting groove of the MHC class 1 molecules. You can see the T cell receptor of the CD8 cell. Okay. So transport of peptides from cytosol to rough endothelial reticulum following digestion by proteasome. This is still a summary of the process involved in the antigen processing and presentation by the MHC class 1 molecule. Peptides are then translocated by TAP into the rough endoplasmic reticulum. Okay. The TAP membrane spanning heteroderma located in the RER, that is the TAP. Okay, TAP1, TAP2, ATP binding casket protein have affinity for peptides, that is, peptides that are between 8 to 6 amino acids, which means the proteins, the peptides, the antigens that are being processed for presentation on the MHC class 1 molecule have this range of amino acid. Then the CD T lymphocytes expresses CD8 receptors in addition to the T cell receptors. Okay, when a cytotoxic T cell CD8 receptor docks, okay, attaches to the MHC class 1 molecule, and the T cell receptor fits the epitope, that is the antigen, within the MHC class 1 molecule, the CD8 lymphocytes trigger the cell to undergo programmed cell death by apoptosis. Okay, so once the endogenous pathogen or the endogenous antigens have been processed and presented on the MHC class 1 molecule of the cell, okay, now this T lymphocyte, the CD8 cells, which are cytotoxic T lymphocytes, are able to attach to these antigens that are being presented by the MHC class 1 molecules. And the next line of action is for them to initiate the death of that cell, because this is a cell, you know, that have been infected either by a virus or whatever, or tumor cells that are out of control. Okay, so the cytotoxic T cells will now attach to the antigens that are being presented on the MHC class 1 molecule, initiate a process of cell death so that the cell will be destroyed, because that cell sort of cannot be remedied because it has been invaded by an organism that have taken over its cell mechanism, its mechanism of action. So the best option for the body is to eliminate such cells, and that is why the cytotoxic T cell lymphocyte comes into play to initiate a programmed cell death. Okay, this helps mediate cellular immunity, which is the primary means to address intracellular pathogens such as viruses and some bacteria that have invaded inside the cell. When you look at a virus like the HIV, when they invade cells, they take over the cell machinery and start, okay, they channel the cell function, the cell properties, the cell machinery, the cell activity to how they want the cell to behave. So the cell became abnormal. So the best bet for the body, the immune system of the body, is to destroy such cell. And that is what is achieved by the MHC class 1 and the cytotoxic T cell receptor CD8. So we also have the major histocompatibility complex 2. Okay. Okay. The MHC class 2 molecules are a class of major histocompatibility complex molecule normally found only on antigen presenting cells. That is cells which are important in initiating immune response. Okay. The MHC class 2 proteins are encoded by the gene of the HLA-D region of the genome in humans. Okay. Anything, property, or any function they talk about the MHC class 2, your mind should go back to what is class 1 doing in that aspect. Unlike class 1 protein, they have restricted tissue distribution and are chiefly found on macrophages, dendritic cells, B cells, and other antigen-presenting cells only. Okay. The antigen presented by class 2 peptides are driven by extracellular protein and not endogenous antigen as in class 1. Class 2 MHC molecule have beta 1 and beta 2 subunits and thus can be recognized by CD4 co-receptors. The MHC class 2 molecule sample extracellular peptides, mainly extracellular pathogens, okay, and by interacting with immune cells like T helper cells, regulate how T cell responds to an infection. So they handle parasites, bacteria that have infected the body but have not invaded the body immune system, the cells of the body. That is what the MHC class 1 molecule handles majorly. So the exogenous pathway or the endocytosis, endocytosis when the macrophages engulf bacteria, okay, from the body. That's the endocytosis process. They are associated with MHC class 2. So exogenous or extracellular antigen is processed and presented by MHC class 2. Okay, the MHC class 2 presents antigen that has been internalized or captured through endocytosis. Once antigen is internalized, it is degraded into peptides within compartments of the endocytic pathway. Peptides are degraded into oligopeptides, around 13 to 18 residues, which then bind to MHC class 2. Then the MHC receptor does not bind with peptide while in the Golgi, but after it leaves and it fuses with endosome. Okay, we have some invariants, some variant proteins that prevent any endogenous peptide from binding, while the class 2 is in the rough endothelium reticulum. Okay, the invariant chain also participates in folding of the alpha and beta chains and direct transport of MHC2 from Golgi to the endocytotic compartment. So there's a mechanism inside in the body to prevent the presentation of endogenous peptides from binding to the MHC class 2 while it is still in the rough endothelial reticulum. Okay, so there's a mechanism by the body system to check which peptides are binding. Okay, so this is also a diagram representing the process of antigen processing and presentation by the MHC class 2. So the invariant chain complexes move from the rough endothelial reticulum to Golgi, then through the endocytic pathway. Okay, so short fragments of invariant chain remains attached clip, prevent premature binding of peptides. Okay, there's a process of specificity to ensure that what it's programmed to address, it's what it's actually handling. Okay, the HLA-DM is a non-classical class 2 MHC molecule located in the endosomal compartment required to catalyze the exchange of clip for antigenic peptide. Once a peptide has bound MHC class 2, it is transported to the membrane. Similar to MHC class 1, peptide binding is required to maintain the stability of MHC class 2. So also from the diagram, can see the antigen presenting cells, the cells that have engulfed the exogenous antigen like bacteria, and it has processed it. After processing it, it's presenting it on its MHC class 2 antigen presenting groove for the CD4 cell to now recognize and destroy the antigen or bacteria. Okay. Sometimes in the immune system, there will be a process that they call cross presentation of antigens. Okay, in certain cases, APC cells present exogenous antigen to CD4 cell by MHC. Cross presentation can also occur. Okay, cross presentation can also occur sometimes, but the classical pathway of presentation of antigen by the MHC class 1, the classical pathway for you to recognize that MHC class 1 presents endogenous antigens, and MHC class 2 presents exogenous antigen. That is the classical pathway, but it also brings to our attention that sometimes they can have cross presentation of antigens. Yeah. So that's just what this slide is all about. So the MHC class 2 antigen presentation pathway, okay, the major histocompatibility complex class 2, the alpha and beta chain expressed by antigen presenting cells. They are expressed by antigen presenting cells. That is MHC class 2 molecule are being expressed by antigen presenting cells. They are synthesized in the endoplasmic reticulum where they form a heterotrimer with the invariant chain. After maturation in the Golgi apparatus, the heterodimer is delivered to the MHC class 2 compartment in which endocytose and exogenous protein, but also the invariants are degraded by proteases for generating peptides. The invariant is progressively degraded into the class 2 invariant chain peptides, which binds to MHC class 2 groove. The chaperone protein HLA-DM induces clip replacement by an antigenic peptide. Then the peptides and the MHC class 2 complex move to the plasma membrane and are presented to the T cell receptors of CD4 T lymphocytes. So you must be able to recognize MHC class 1 molecules, the cells that present or express MHC class 1 molecules, the cells that present MHC express MHC class 2 molecule. The classical view How does the MHC class 1 molecule what antigen does it process and present? The MHC class 2, what antigens? Is it endogenous or exogenous antigens? So the classical pathway of their function. That's why I didn't want to try to talk much on the cross presentation aspect of it. So they suffice it for now for you to be able to differentiate the classical pathway of cell presentation by each of these MHC molecules. Okay, we say that antibodies can recognize antigens alone. T cell receptor can only recognize antigens that has been processed and presented by the major histocompatibility complex. This required antigen processing, antigen presentation. Discovery of the MHC complex. The MHC complex was initially discovered as a genetic locus associated with the acceptance or rejection of transplanted organs in mice by Gregor Snell in the 40s. In the 1950s, the same genetic system was described in humans by Jane Dossett and Jane Van Rode and was called human leukocyte antigens. This is as a summary of what we have stated earlier. Okay, these are MHC genes, binding of polypeptide to MHC molecules, that is antigen presentation, MHC polymorphism. Okay, so that is, you have to just know the classical pathway of MHC class 1 molecule presentation, processing and presentation, the classical pathway of the MHC class 2 processing and presentation of antigens to the T receptor cells. In as much as the antibodies can recognize antigens, for the T cell to participate in the immune system of the body, the antigen has to be processed and presented on the MHC molecules. That is how the cytotoxic T cells, the T helper CD4 cells, can help in the body immune system. And we say that the MHC molecule, remember, plays role in the adaptive immunity of the body. Okay, let us look at the biological significance, the importance of these MHC molecules. The HLA, as it is called in humans, is very, very important in the sense that it serves as a barrier for hematopoietic stem cell transplantation. Okay, therefore, HLA matching and compatibility between the donor and the recipient is necessary for a successful transplants. HLA antigen also have a role in graft versus host disease, a potentially serious complication of allogenic stem cell transplantation. So, in essence, we are saying that the HLA system, the HLA complex, plays a very, very significant role, which I've already said earlier, plays a very, very significant role in hematopoietic cell transplants, hematopoietic cell transplants, because there must be a compatibility in matching of the HLA system of the donor. There must be compatibility, the HLA system of the donor and that of the recipient. So it's not a complex that can be ignored. It's a complex that determines whether the transplant, like hematopoietic stem cell transplants, they can do, sometimes they do a sickle cell disease patients, okay, and some other hematological malignancies where they need to transplant stem cells. So you find out that the HLA cross-matching and compatibility is very, very important in that aspect. Also, in solid organ transplantation, HLA antibody play an important role. With patient exposure to foreign HLA antigen due to pregnancy or blood transfusion or transplantation, they can make antibodies against epitopes of those foreign HLA molecules. This is called HLA alloimmunization. Presence of HLA antibodies against graft can cause antibody-mediated rejection and graft loss. Okay, also in solid organ transplantation like transplanting of a heart, kidney, okay, the HLA system also plays a very important role because some individuals, due to pregnancy or blood transfusion or transplantation, have make antibodies against some of these HLA antigens. Also, the HLA plays a role in alloimmunization, can also cause platelet refractoriness. This condition creates difficulty in finding compatible platelet units, especially in transfusion-dependent HPC transplant patients. So, in such cases where platelets transfusion does not amount to increase in platelet number due to the alloimmunization that's occurred through the HLA system. Also, HLA antibodies also play a role in febrile non-hemolytic transfusion reactions and transfusion-related acute lung injury. Truly, we'll see some of these disorders as we go along, but we have also encountered that in our fourth year. So we're going to look briefly on HLA and transplantation. Transplantation can be solid, can be transfusions, can involve the fluid parts of the body. So HLA is very, very important when we are discussing transplantation, either solid organ transplantation or hematopoietic stem cell transplantation. HLA is also important. We are discussing transfusion therapy. Okay, it can give rise to platelet transfusion immune refractoriness, TA transfusion associated graft versus host disease, febrile non-hemolytic transfusion reactions, transfusion related acute lung injury. Okay, HLA, there's a connection also between HLA and some diseases. In paternal testing, it's the HLA complex that is being characterized. So the importance of the MHC polymorphism, whether MHC or HLA, we are still referring to the same complex. So the MHC genes are highly polymorphic. This means that there are many different alleles in the different individuals inside a population. The polymorphism is so high that in a mixed population, there are not two individuals with exactly the same set of MHC genes and molecules, with the exception of identical twins. So the MHC complex is a highly polymorphic complex. Two individuals have the same set of MHC genes and molecules. The evolution of the MHC polymorphism ensures that a population will not succumb to a new pathogen or a mutated one, because at least some individual will be able to develop an adequate immune response to win over the pathogen. I've also emphasized on this earlier, the advantage and the disadvantage of this complex. The disadvantage lies majorly in the part that it makes it difficult during transplants, like solid organ or stem cell transplants, makes it difficult to find a match for an individual. Be that as it may, its advantage lies in the fact that it helps the population to be able to mount immune response to any new pathogen or mutated one that can in situations of epidemics. Okay, the population will not be wiped away, because at least some people will be able to mount and resist the new pathogen that have invaded the population. It's where the MHC molecule plays a significant, positive, significant role. play the main role in graft rejection in distinguishing self from non-self. Okay. Connected with autoimmune disorder and other diseases, e.g. type 1 diabetes. Okay. So when we talked about transplants, we have different types. It can be autograft, okay? Graft in the same person from one part of the body to other area of the body, but within the same individual, as you can see in the diagram. We have isograft between identical twins, okay? So we can have also the most common one is the allograft. This is between different members of the same species, like from one human to another human being. You can transplant kidney, can transplant the heart, can transplant lung, liver, whichever organ that is being transplanted from one person to the other individuals. Or what is xenograft? I don't know whether this exists between two different species, likely monkey to humans. I don't know how that still exists. Okay. So solid organ transplant. Various organs can be donated by either deceased donor, somebody that is late already, before the person gives up, or at the tail end of life, the organ can be harvested from the individuals, can be donated by living related donors, which are also very common. Okay, can also be donated by living non-related donors. Living donors are mostly for kidney transplants, of course. So it can be from a deceased donor, a related donor, non-related donor. Okay. The impact of HLA mismatching and allosensitization on organ transplants. Antibodies bound to the graft fix complements and cause damage to the vascular endothelium, resulting in thrombosis, platelet aggregation, hemorrhage. So when there is a mismatch of any organ from one individual, probably a kidney from one individual to another person is being transplanted, okay? We have some antibodies that are found on the graft, okay? These antibodies can fix complements. When they fix complements, when complements are fixed, the coagulation pathway can be initiated. And when the coagulation pathway has been initiated, we have what we call thrombosis. Okay, we did that very well in our fourth year. We have thrombosis, we have platelet aggregation, we have hemorrhage. All this process will tend to cut off the organ that is being transplanted from getting the supply of blood. By denying the supply of blood, of course, the oxygen and the nutrients are also being denied to this organ, and it eventually dies off. So they say that the transplant failed, okay, because of this process of coagulation that is being initiated. Once thrombosis is being set up, there's a block in the flow of blood. Blood cannot reach the organ that is being transplanted, okay, or hemorrhage or whatever. All this leads to cutting off the new organ, okay. So there are mechanisms for this rejection. This rejection can be very, very hyper acute, very, very immediate, or it can be acute, okay, or it can be also chronic. Acute, usually cellular rejection, can be reversed by increasing immunosuppression. That is why sometimes these patients that received organ transplants, okay, they tend to give them medications and treatment therapy to suppress the immune system. However, the immune system is trying to reject the non-self. They tend to give therapy in such a way to subdue the immune response of the recipients of that organ. Okay, so acute rejection can be handled by increasing immunosuppression of the recipient. Okay. Rejection can be chronic, combination of cellular and antibody mediated rejection. So the process of organ rejection can be very, very acute, or just acute, or can be chronic, depending on the level of mismatch of the HLA complex. So mechanism of rejection, we have the hyperacute rejection, as we say, occurs between minutes to hours after transplantation. Patients already have antibodies specific to a graft, okay? Just like we talk about in our blood transfusion, okay? Okay. Some things that can give rise to this mechanism of hyperacute rejection is the natural ABO expressed on endothelial cells and preformed HLA antibodies, okay? Characterized by arterial and glomerular thrombi caused by damage inflicted to the endothelium. Preformed anti-HLA antibodies activate complements, bind to the endothelium, leading to the release of von Willebrand factor, resulting in platelet aggregation, irreversible ischemia, consequent allograft rejection. So the prevention is by ABO identical transplantation, confirming negative lymphocyte cross-match. The treatment is just to remove the organ because it has been cut off from the supply of by the recipients. So where there's a transplant that is not ABO compatible can lead to this hyperacute rejection of transplanted organs. And as much as they are matching the HLA system, they also match the ABO blood group system of the individuals involved. So we have acute rejection. It occurs from 1 week to 3 months after transplantation. Disease incidence as a result of the introduction of immunosuppressive. We have, decreased incidence of this acute rejection due to the use of immunosuppressive drugs. It can be either cell mediated, okay, 90% of it, CD8 cytotoxic cells that recognize class 1 are the main effector of this rejection, okay, or antibody mediated, about 20 to 30% of it are antibody mediated. They may have a humoral component, okay, there will be a pre-sensitization. The immune response is mostly mediated by de novo donor-specific antigen against HLA, endothelial cell antigen, and ABO antigen. The DSA is activated by the complement, resulting in endothelium injury and consequent graft rejection. So already the recipients have some antigen specific against the HLA, so that when the tissue or organ is being transplanted, it initiates complement fixation and the subsequent reactions that follows. This can be handled by plasma exchange and intravenous immunoglobulins, okay, lymphocyte depleting antibodies, and all that. So we have the also chronic rejection is an array of chronic transplantation disorders that occurs months to years after transplantation without a specific etiology. It's low cell mediated rejection, low donor specific antigen titers, or other recurrent non-immunological disease are all potential causes. So in hemopoietic stem cell transplantation, where we have allogenic HSCT established as a mode of curative therapy for hematologic malignancies such as severe aplastic anemia, severe congenital immune deficiency, selected inherited metabolic disease, okay, So in some of these hematological malignancies, hemopoietic stem cell transplantation has been established as curative for some of these malignancies. Then the source of the hematologic stem cell can be from bone marrow, mobilized peripheral blood stem cells, umbilical cord blood. These are sources of the hemopoietic stem cells. Okay. The HLA can elicit an immune response either by presentation of viable peptides or by recognition of polymorphic fragments of foreign HLA molecules. And if there's any mismatch in this process, it can lead to graft failure, delayed immune reconstitution, graft versus host diseases, and death of the recipients. Okay. The most important loci influencing post-transplant outcome of patients giving hemopoietic cell transplant from unrelated donor are the HLA A, B, and C DRBI loci. The best compatible hemopoietic stem cell are from identical twin and a genotypically HLA identical sibling. Prevention of such uh, complication is by leukoreduction. Avoid blood products from relative or transplant patients. Okay, we'll see the details of these disorders as we go on, taking them one after the other, the graft versus host disease and the non-hemolytic transfusion reaction. Okay, HLA in transfusion therapy. Okay, it's still significant because it can elicit some clinical conditions in the patients. Transfusion or transplantation of blood components bearing allogenic HLA can stimulate clinically significant immunological response. All cellular components except erythrocytes express HLA, and any plasma containing product may include HLA-specific antibodies, which are potentially harmful to the recipient. So even in blood transfusion, HLA plays significant role. Why? Because during blood transfusion, some white cells can still form part of the red cell blood cell transfusion, and this white cell expresses HLA antigen, which can elicit some reactions. And also, even if it is plasma product that is being transfused, some of them can also have HLA specific antibodies. And during ordinary routinely in our geographical settings, during ordinary blood transfusion, we don't really cross-match for HLA antigens. We only cross-match for the red blood cell antigens, okay? So because of that, sometimes after transfusion, find out that the patients come down with some clinical conditions due to these HLA antigens and antibodies. So in transfusion, HLA can actually cause platelet transfusion refractoriness. It can cause febrile transfusion reactions. It can cause transfusion-related acute lung injury. It can cause transfusion versus host disease. Okay. So we handle some of those. Okay. So platelets transfusion refractoriness. Platelet transfusion refractoriness, or rather we may cap it here today so that next time we will now handle all those. Okay, so